A bit, I guess.
Just a touch, right? That's amazing. Okay, so what I was saying was that the low energy electrons here and the low energy electrons here understand the cycling here. All right, the electrons are pulled out, they're excited, right? Then the excited electrons are pulled out again. The energy is removed from them, creates a proton gradient, fine. And then these low energy electrons are bonded to the oxygen, and that's why water has low energy electrons in it. The water that photosynthesis consumes is a byproduct of cell rest. Right? That's where you really start to understand the cycle, in that the water that photosynthesis requires is the byproduct of cellular respiration. The same low energy electrons that are pulled out of this water by photolysis here are the same low energy electrons at the end of the electron transport chain that you'll find in cell rest. So why do we breathe oxygen? We have to breathe oxygen to pick up the low energy electrons at the end of cell rest. We then stabilize these electrons by manufacturing water. So that means plants need oxygen too. Another huge misconception. People think that only animals need oxygen. All organisms need oxygen except bacteria, and bacteria play metabolic games. It's like we make all these rules for eukaryotes, and then we say this applies to almost every life form on Earth except the prokaryotes. Right? The prokaryotes make ATP differently. The prokaryotes do cell rest differently. Prokaryotes do cell division differently. There are minor differences in their DNA replication. Basically, all the major rules that we talk about apply to eukaryotes, and prokaryotes do kind of the same thing, but always in a different manner. Always. Okay. So when you walk up to a child and say, you know, what gas do you have to breathe or you'll die? And they say oxygen. Now you can explain to them why. You can say, well, there's these electrons, you see. And you will be amazed if you decide to have children, and there's plenty of time, that you can teach them some really upper level science when they're small. And they haven't been shown to be afraid of it yet, so they're fearless. It's really, really cool. All right, so without color, let's write the balanced equation here. So we've got 6 carbon dioxide plus 6 water. How about if we do that in green? That's much better. Then I'm going to ignore the whole enzymes and light, and then we've got C6H12O6 plus 6 moles of oxygen. Right? Good old chemistry. Then you come down here and you say 1 mole of glucose plus 6 moles oxygen. It's the same reaction, just backwards, right? Then 6 moles of CO2 and 6 moles of water. And then per glucose, you produce 36 ATP, like such. So there is the balanced equation. What I want you to focus on is the energy that comes in here, and it doesn't come in directly to the carbon dioxide, of course, and then the energy that comes out here. And I need to mute my phone. Probably a freshman saying, I texted you at 11 o'clock last night, Mr. Manra, and you didn't respond. Yep. Oh, that's hilarious. How dare I not respond when they text me at 10 o'clock at night? So, energy in, chemical energy out. So then come up here. So you've heard the saying that all of us ultimately run on sunlight. Now you know the chemistry as to why. Okay, so you come up here and you have light energy from the sun that's captured by autotrophs. And then ultimately that energy comes out here as chemical energy in the form of ATP. Okay. So the energy that you are burning right now, you're burning energy to make your heart beat, as your cells do their cellular work, as you're breathing, as you're thinking, as the muscles are pulling your eyes in different directions. The muscles are in your arm are contracting so that you can write or so that you can type. All of that is consuming ATP. The energy that's in the ATP that you are burning right now ultimately came from where?
Sun.
The sun. Okay, there's an energy transfer. So the energy comes from the sun into autotrophs, into a heterotroph like you. You then break that bond in the ATP and you convert that energy into motion, light, heat. Maybe you make other bonds as you synthesize a molecule. Maybe you use that energy to power an enzyme to break down other molecules. You consume that energy metabolically, right? So the energy flows through you and ultimately back out into the universe. Energy does not recycle. It flows through us from the sun, through the food web, and out into the universe. But ultimately, from our perspective, it's sunlight to ATP. We all run on the energy from the sun. This is why it was such an issue at the end of the Cretaceous when that meteoroid hit the surface of the Earth, right? And blocked out the sun for a certain period of time. And that caused, it almost wiped out all life on Earth. It was close, right? Well, why was that an issue? Because there was no sunlight to excite electrons. Thus, the ecosystems were deprived of tremendous amounts of heat. Now, the one group that was hit the hardest were, of course, the dinosaurs, the reptiles, right? And so why? Well, that's a heat issue because they're non-homeotherms. They don't make their own heat, whereas mammals, we make our own heat, so we were successful. It's good to be a mammal. Wasn't at that time, but it is now. So ultimately, the ATP is used for cellular work. What that work is totally depends on your needs. So when we're talking about cell resp, this is all about ATP. It starts and ends, it's all about ATP synthesis. Photosynthesis is all about producing G3P. That's really what photosynthesis is all about, whereas cell resp, it's all about the ATP. So you have to be comfortable with the cycle of ATP and ADP. Now, in biology, to tell the difference between phosphate and what is just phosphorus, see, biologists drive chemists nuts because all the time as a biologist, we write P and we mean phosphate. But a chemist sees that and they see phosphorus. And so they're like, you biologists are so sloppy. And we go, you chemists are so technical, right? It's really the way it works. So you will see people commonly write a little I, okay? It's a subset I. It's not PI like pi, like the math. It's P with a subset I that stands for inorganic phosphate. And this is a biologist's footnote letting the chemist know that this is phosphate and not a phosphorus. Fine. So it's ATP, and then you will hear people say ADP and an inorganic phosphate. And this simply cycles this way, and then it cycles back. Just bounces back and forth. Super easy. Okay. So energy comes out, energy goes in. So energy comes out, energy goes in. Such. Go over there. So energy comes out and energy goes in. Okay, so energy comes out. We all know that ATP is the energy molecule for all living things. Cool. So when this energy comes out, this is energy out. Why? To do work, whatever that cellular work is. Okay, because it turns out that the adenosine then has a phosphate, and a third phosphate. So between the second and the third phosphate, there's a very, very high energy bond. And when that bond is broken, that releases energy, and that then is what the cell uses. So that's why you then break off this inorganic phosphate. Cool. So then energy has to come in here. So this is energy in. Now, we're not talking photosynthesis here, we're talking cellular respiration. How does the energy come into this cycle? This is energy in from where?
Macromolecules.
Macromolecules, you bet, from food. Love it. High energy electrons in macromolecules. This is why if you don't eat, you starve to death. You actually run out of energy. Cycles back and forth. Energy then goes in. That powers the phosphorylation of ADP and P, and you're back to where you started. Cool?
Yep.
May I take it? Anybody need it? Okay, just like when we did photosynthesis, we have to make sure that you're comfortable where we are in the cell. Okay, so let's draw ourselves a cell. Cool. And you, of course, then have the nucleus, DNA, like such. Okay, so you have plasma membrane. And let's go from the outside, nucleus. Awesome. And then you have kind of a kidney bean shaped, it's commonly drawn as a kidney bean. And this is the mitochondrion. Now, what makes the mitochondrion interesting is that it's also a double membraned organelle. Like that. So there's an inner membrane and an outer membrane. Cool. So we always say that cellular respiration happens in the mitochondrion. Kind of. Okay, for the most part. Okay. So cellular respiration, I can slide that in. Cellular respiration has 3 steps. 1, 2, and 3. Okay. The first step is called glycolysis. Glyco, sugar, lysis, split. The first step is literally called the splitting of sugar because that's what happens, right? You take all of your sugars, which are nothing more than carbon rings, 5 to 6 carbons, and those rings are simply cut in half. And then they're now a segment of carbon chains, and then now you start pulling the electrons out. The second step is named after the gentleman who discovered it, Dr. Krebs, therefore it's called the Krebs cycle. Krebs is Calvin backwards, okay? Calvin makes G3P and bonds high energy electrons to substrates. So Calvin is very much a reductive process. Krebs is the opposite. Krebs takes carbon compounds, hydrocarbons, with high energy electrons in them and pulls the high energy electrons out. So while Calvin is a reductive process, it reduces substrates. Krebs is a highly oxidative process. Krebs is pulling those high energy electrons out. And then of course it ends with an electron transport chain, because if you're going to pull all of the high energy electrons out here, right, then those high energy electrons are delivered here. And gee, I wonder what's going to happen, right? You're going to have an electron transport chain. What's going to be pumped?
Protons.
Protons. That's going to make a proton gradient. And then, gee, which enzyme?
ATP synthase.
ATP synthase will then have those protons flow through the ATP synthase by chemiosmosis, and the mitochondrion produces large amounts of ATP. Huh, sound familiar? Right? This is extremely similar to photosynthesis, except the ATP that's being produced here is in a much, much higher concentration to power all of the metabolic processes in the cell. Okay, so there's 3 major steps to cell resp. Now, some of your professors will teach it as 4 steps. All right, there's a reaction in here that links glycolysis to Krebs, has a really fancy name. It's called the link reaction, okay? And depending on how your professors teach it, IB groups Krebs and the link reaction together, okay? So that's why I'm teaching you that cell resp has 3 steps. However, you might have professors that separate the link reaction out, and so you will hear them say cellular respiration has 4 steps: glycolysis, link, Krebs, and the ETC. Fine. In their class, then if they want 4, give them 4. Also, I teach the traditional 36 ATP, depending on different molecules are shuttled throughout this process. And so as ATP is consumed, some people will say as few as 32 ATP are produced. If you ignore all of that shuttling, you can get up to as high as 38, sometimes 40 ATP. So there's a range depending on how specific you want to be on the chemistry. So find out what your professors want and give it to them, all right? If you get a 36 professor, give her 36. If you get a 38 professor, then on his exam, give him 38. Whatever the professor asks for, give it to him. It just depends on how much depth they're going to teach you, okay? But that's why there's variation. All right, so we say that cell resp happens in the mitochondrion for the most part, okay? Glycolysis happens out here. Glycolysis happens out here in the cytoplasm. Okay, so glycolysis is a cytoplasmic process. And all this is the splitting of the carbon rings in sugars into a chain. That's all it is. Simple, okay? Then the products of glycolysis are then, here's your shuttle, are shuttled into the middle, the center, the core of the mitochondrion, okay? And the reaction that happens from the products of the glycolysis to the center of the mitochondrion, this is called the link reaction. Okay, some people couple this with Krebs and some people don't, just depends, right? So you then have this oxidative reaction here where the high energy electrons are removed. So this is the Krebs cycle. The Krebs cycle happens in the core, in the center of the mitochondrion. Okay, so that would be step 2. So I should get step 2 like this. That's probably tiny from your perspective. Let's make that bigger. Ah, there we go, much better. Like such, okay? So if Krebs is oxidative, then what is being ripped out of all the substrates that go through Krebs? What is being pulled out?
Electrons.
Electrons, okay? So what do you think then is embedded in this inner membrane that receives all of those electrons? We think sits just like that, where all of the electrons then go.
Enzymes.
Proteins. Not exactly. They're carrying the electrons.
Ribosomes?
Ribosomes, that would be on the rough ER. If you think that high energy electrons are ripped out, right? This is step 1. This is step 2. What is step 3 in cell resp? There you go. From way downtown. ETC. So cool. Just like this. Electron transport chain. The electrons are then ripped out here and delivered to the electron transport chain. Electrons are transported between these different proteins. Protons get pumped. You then get a gradient. And then what enzyme sits over here once there's the proton gradient?
ATP synthase.
ATP synthase. Not considered a step in cellular respiration, which I think is interesting, right? You make the proton gradient, oh yeah, and then you make ATP. So phosphorylation is not considered a formal step of cellular respiration, which I think is interesting. So when we say that cell resp happens in the mitochondrion, for the most part, the majority of it occurs inside of the mitochondrion. Okay. Comfortable?
Yes.
Okay. All right. And let's do one more overview picture, and then that's where we'll stop for today. And then we will get into the specific handouts on Thursday. Okay. So let's put a box here. Such. And let's put a slightly bigger box here. And let's put a big old box over here. What do you think those 3 boxes are? I'm sorry?
The steps.
Totally. So what goes here?
Glycolysis.
That's glycolysis. How about here?
The Krebs cycle.
And
ETT.
ETC, I agree. Okay. 3 major steps. It's just a flow is what this is. It's a story. Step 1, step 2, step 3. Fine. Now, always to keep our chemistry straightforward, let's use glucose. 1 glucose. How many carbons in glucose?
5?
6.
6.
6.
Yeah.
6, I agree. Okay. 6 carbons. There's a 5 carbon ring, and then it has a carbon off of one of the central carbons. That's where the whole alpha and beta glucose come from. Fine. So inside of glycolysis, some of the electrons start getting pulled out, which means glucose goes through what process as the electrons are pulled out of the glucose? What do you call it when you pull electrons out?
Oxidation.
That's oxidation. Okay, so all the chemistry here and here is all oxidative. It's all oxidation. The high energy electrons are pulled out. Okay, cool. So the high energy electrons are pulled out here, but they need to be delivered here. So what do we need in between these 2 steps? An electron carrier. Totally agree. The electron carrier in cell resp is NAD. Not NADP, it's NAD. There's no phosphate in it. And so students will then say, well, how do I remember NADP and NAD? Remember that NADP is in photosynthesis, and photosynthesis starts with a P. All right, so NADP is in photosynthesis, NAD is in cell resp. Fine. So NAD comes over here and picks up those high energy electrons and delivers them over here, and then comes back like such. So when NAD and its associated proton picks up the electrons, what does NAD become? Goes right there.
NADH.
NADH. NADH is then carrying the high energy electrons, right? So here's your high energy electrons. The NADH then drops off the high energy electrons and goes back to being NAD, and it just cycles back and forth and back and forth. It's a truck driver is what it is, right? Truck drivers pick materials up, bring them here, drop them off. That's what they do for work. So then what do I need to label that arrow right there as NAD gains the electrons?
Reduction.
That's reduction. Oxidation reduction. Simple. So glucose, it's a ring, 6 carbons. You then take that 6 carbon compound and you break it in half. So you go from a 6 carbon ring to 2 3 carbon segments. Okay, so from 1 glucose, you break this apart and you get 2 3 carbon compounds. This 3 carbon compound is called pyruvate. Again, don't be intimidated by terminology. Just because it's unfamiliar doesn't mean it's difficult. It's just unfamiliar. Fine. So 6 carbons all together, glucose. Rip this in half, pyruvate. Now, do you think there might be a little chemistry that I'm overlooking right here? Just a little. Yeah, there's 10 steps here, all right? And if you're a bio major, you will take biochemistry your junior year, and your professors will make you memorize those 10 steps. It's not difficult, okay? Seriously, it's memorization, okay? And most of us are pretty good with memorization. And you memorize it, and you take a test, and you do really well in the test, and they go, good job, and you go, thank you, and then you know what you do with that information? You forget it. Okay, because nobody walks up to you and goes, what is the 6th step in glycolysis? But it's like this rite of passage for all biochemistry classes that you have to memorize the steps. You memorize the steps. Take the test, you'll be fine. Okay, so 6 carbon ring, break it in half, 2 3 carbon compounds. Fine. We then get into Krebs. Holy cow. You had a little bit of oxidation here, but guess what? A lot of oxidation happens here. A lot of it. Okay. And we'll diagram that on Thursday. Okay? Lots and lots and lots of oxidation. However, also here, CO2 is ripped out. In fact, all 6 of your CO2 are ripped out here. It's actually 2 in the link reaction right there and 4 here. That detail is important. All 6 CO2 come out. So all of the CO2 that we breathe out, 2 of them are pulled out right there in what's called the link reaction. If you really want to know, the link reaction sits right there. 2 of the carbons are pulled out here, and 4 of the carbons are pulled out here. So as you sit here taking your notes, CO2, right? Where'd all that CO2 come from? 1/3 of it comes from here, and 2/3 of it comes from there. So as the CO2 is pulled out, what do we call that process when CO2 is removed?
Decarboxylation.
Totally agree. Decarboxylation, right there. The CO2 you breathe out is due to the decarboxylation that happens during link and Krebs as you decarboxylate the food that you eat. Simple. But there are a whole bunch of electrons being pulled out. So NAD comes in, grabs those electrons, leaves, drops the electrons off, and comes back. NADH. There's then an electron carrier that instead of using nicotinamide, it uses flavonoids. You don't have to worry about that. But it changes the name of the electron carrier. High energy electrons are pulled out here, and high energy electrons are pulled out here. Boom, just like that. So you have electron carriers, NAD, NAD, and FAD. And all they do is they pick up the high energy electrons that are oxidized from your food, and they deliver those electrons, a lot of them, to the ETC. There are a lot more electrons delivered to the ETC, which is why cellular respiration produces so much more ATP than the electron transport chain and ATP synthase of the photosystems in the thylakoid. Okay, so what do I need to put there and there?
Reduction.
Reduction. Oxidation reduction. Chemistry. Be comfortable with your chemistry. As bio majors, you're going to take a lot of chemistry. If you can, over the summer or any time, any bridge programs, if you're going to be a bio major, I'm telling you, chemistry. Okay, students don't come back and complain about their biology prep. Okay, what they do is they complain and they come back and they go, oh man, chemistry. Tell the bio majors chemistry. You got it. So guess what? Bio majors, chemistry. All right, focus on your chemistry. It's important. So what you end up with is a tremendous amount of high energy electrons building up here in your electron transport chain. Through something that we'll talk about on Friday, those high energy electrons are converted to low energy electrons. It's not fancy. You just haven't talked about it yet. And what comes out?
ATP.
Almost.
Energy.
Energy, okay? That's the whole proton gradient chemiosmosis kind of thing. All right. Then we need to talk about that energy is then used for? Yep. To make ATP. A whole bunch of it. That's not bad. What do we call this process of bonding an inorganic phosphate to ADP?
Phosphorylation.
Excellent. That's phosphorylation. Phosphorylation. You need to get rid of these. How do you get rid of these? Well, you first have to bond that electron to what?
Electron pair.
Not a, well, kind of a carrier. It's an electron receptor or acceptor, right? But we need to let me ask a better question. We need to convert that electron to hydrogen, not a proton, hydrogen. How do you make an electron into hydrogen?
Bonding a proton to the electron.
Totally agree. You bond a proton to it. An electron bonded to a proton makes hydrogen. This is not a proton. See, this is where chemists confuse us, because a proton is written H+. That is a proton. Or then they really confuse us and they call this a hydrogen ion. You go, oh, come on, right? It's a proton, okay? This is hydrogen, the atom, hydrogen, the element, right? All right, but you can't let this hydrogen build up. You can't let these electrons build up, because if you let these build up, then the carriers can't bring any more electrons. If the carriers can't bring any more electrons, then that shuts down ATP synthesis, okay? So you have to remove these from the process. How do you do that? How do cells remove the hydrogen at the very end? Bond it to oxygen to make?
Water.
Water, H2O. And it's actually 4 protons for every oxygen molecule. There's a lot. We should actually talk about that. So ultimately, what happens to the oxygen here? As it's picking up these electrons?
It's reduced.
It's reduced. This is the only time you'll ever see this by itself. Even in photosynthesis, the water gets oxidized and the NADP gets reduced, right? Then you go into Calvin, the NADPH gets oxidized and BPG gets reduced, right? And I said they're always coupled, except for here. there's just this reduction just kind of hanging out. And you say, well, okay, but this really isn't isolated by itself, because where's the oxidation? If reduction is here, where's the oxidation? Because these are always coupled. Where's the oxidation?
In photosynthesis. Oh.
Yeah. Why should we write oxidation here?
Since the electron transport chain is gaining the electrons.
Yep. The electron carriers are giving up the electrons and the chain accepts them. Yep. So oxidation here, reduction at the end. So ultimately, we have to breathe oxygen in order to pick up the low energy electrons at the end of the electron transport chain. So when someone is deprived of oxygen, really unfortunate, right? But if you deprive them of oxygen, right, then that means the electrons build up until eventually the electron transport chain shuts down. The electron transport chain shuts down, no proton gradient. No proton gradient, no phosphorylation, and ATP synthesis stops, okay? And the first cells that start to die without ATP are neurons, brain cells. It takes about 6, 7, 8 minutes. Okay, that's why when someone is deprived of oxygen, brain damage starts after about 5, 6, 7, 8 minutes. Okay, and then that gets really serious really fast, because that's where they have to decide, you know, we can resuscitate the person, we can revive them. However, you might revive them with significant brain damage, and while you brought the body back of the individual, you may not have brought their full mind. And that's where doctors have to make the decision, or the family member has to make the decision to stop the resuscitation. Really, really heavy duty, serious decision. Okay, so what we're going to do then on Thursday, that won't be as serious, is we will take these diagrams and we'll simply break them down, right? And so if you've looked at any of the handouts, right, we'll start here. G, step 1, step 2, step 3. Look familiar? Okay, and then do you think there's a handout for each one? There's glycolysis, 10 steps, right? They're so pretty in color. Then there's the link reaction, right? And then do you remember when we counted carbons in Kelvin? What do you think we're going to do here?
The same.
We're gonna do the same, okay? It's not as complicated. It's not like Kelvin's hard to get across, because you have to show one molecule, but there's 3 of each, boys and girls. So we show you 5, but it's really 15. We show you 6, but it's really 18. Krebs isn't that complicated, right? You have 4, and 4 plus 2 is 6. And then 6 minus 1 is 5, and then 5 minus 1 is 4, and it's 4, 4, 4, 4, 4. 4 plus 2 is 6, and then so it's much more straightforward. And then after Krebs is the ETC. Look familiar? Electron transport chain, flow of electrons, pumping of protons, proton gradient, chemiosmosis, phosphorylation. Okay, we can actually go through cell rest much smoother than we did photosynthesis, first of all, because the Krebs cycle is much more simple, and that you don't have to get across this idea of 3 molecules at the same time. And now you have a background of electron carriers, oxidation reduction, and the electron transport chain. And this goes much, much quicker. So we will finish that this week. And then we'll start talking about presentations the week after that. And we might get to 8C, maybe not. We'll see how it goes, okay? I also still have not heard about your graduation, but I might, I will. I'll drop Mitrovich a note and say, hey, what's going on with the grouping during graduation? And of course, as soon as I hear, I will let you know. I'm actually going over to Chula High. I'm going to join Aiden and Isabella. We're doing recordings on Friday, and so they want me to do the announcement, as I always do at graduation, of all the IB names. I don't know if that's a backup, or if they're going to make a video, if they're going to play it on this. I have no idea. They said, hey, IB coordinator, get your butt over to Chula High and read some names. Yes, ma'am. So that's what I'm doing on Friday. We'll figure out once I get there. And then what else? We talked about the vaccine. We talked about the vaccinations at the school. Don't forget about that survey for me, or for us. Ultimately, it's for you. And then the core and exam by Thursday.